Forward-looking statements. We are a publicly traded company on the NASDAQ. So we, at our heart, are a technology company. We were founded by 5 scientific founders in virology, and we spent our early formative years developing that technology, building a broad base of IP, and deciding where to apply that technology. And where we're applying it is in ophthalmology. And we have a broad portfolio within the ophthalmology space that I'll discuss. And we, last year, completed a key collaboration agreement with Biogen. This is our pipeline, and it's just the top of our pipeline, the lead programs. And what you see here is that we have licensed XLRS, X-linked retinoschisis, and XLRP, X-linked retinitis pigmentosa, to Biogen, our partner, but then we have retained inside the company additional indications. What I want you to get out of this is that while we're operating in the orphan ophthalmology space, these lead indications are significant commercialization opportunities in that they have 20, 30, 40,000 patients in the US and Europe combined. So we have created a very flexible platform, and since we started out as a technology-focused company, what we've developed is a very robust capability to design these vectors specifically for each and every indication that we're working on. And for each indication, the capsid might be most important. For another indication, it might be the promoter. For yet another indication, it might be the formulation. But all of these need to be designed together to specifically address the indication we're working on, and that has also allowed us to build this rich and robust IP portfolio. So why ophthalmology? This is probably a slide I could eliminate for this group, but what's most important and what intrigued us the most about moving into the ophthalmology space was the fact that there'd been such a great deal of research done because sight is so important to human beings, and we had very good animal models to screen our products, and we had a large set of endpoints, both approvable endpoints and surrogate endpoints to look at as we develop products going forward. So again, this is a slide that most of this audience will be familiar with, but I want to draw your attention to what will be your right-hand side, just to illustrate in a very colorful way how specific the retina is. And for each of the genetic mutations that we're working on, you really need to understand exactly where in the retina do you want to get. And that's how you design your product, the combination of the delivery, the capsid, and the promoter, so you can get to that specific place. Our lead program is X-linked retinoschisis. Again, this was an indication where there was a good animal model, there was initial proof of concept data, we completed all the IND-enabling steps, and we're now in the clinic. This is a standard orphan drug development phase 1-2, and that it's a dose escalation study in adults, and then an expansion cohort that will include children as young as the age of 6. It is primarily a safety study. However, because we're in patients, we're able to get early signs of efficacy. This trial is ongoing. We've dosed 8 patients. The product has been very well tolerated with only mild to moderate inflammation that's either spontaneously resolved or been treated with steroids. And we've expanded to now 7 sites across the United States so that we can move forward and complete the trial efficiently. One of our next lead programs are 2 different treatments for achromatopsia. Achromatopsia is a very significant indication within ophthalmology in that all of these patients are legally blind. Again, there was a very good animal model, a large animal model in the dogs where it showed very good data, and we were able to complete all those IND-enabling studies and move into the clinic. So we are in the clinic with the first of the 2 products for achromatopsia. Again, it's a dose escalation study with then an expansion cohort to include children. And the primary endpoint is safety. The product has been well tolerated to date. 2 patients have been dosed, and we'll be reporting data on this trial when we have significant data over enough patients and enough time points to be able to interpret that data accurately. The next of our lead programs is a treatment for X-linked retinitis pigmentosa. So this is probably a disease many people are quite familiar with. Again, there is a large animal model. It's a naturally occurring dog model, and we have published data in that dog model showing that treatments with gene therapy vectors can arrest the macular degeneration and preserve photoreceptors, which you can see in the side there at the bottom. We're currently completing the IND-enabling studies for this indication and will be in the clinic next year. So we do have a very experienced team that we've put together. As I said, we're very technology-based, very data-driven, and we've put together a team that has experience not just in virology, but also ophthalmology. And as we've expanded our company over the last 2 years from 12 people to 65 people, we've remained dedicated to the idea of bringing on highly experienced people, whether it be in CMC, virology, clinical development, and clinical operations. Financially, we're very strong. As we reported for our June 30, which is our year end, we have $173 million of cash on hand, and we expect at our next year end in June 2017 to have between 100 and 120 million. That's at least 2 years of cash, and it provides us all the resources we need, combined with our talented team and the key opinion leaders and all of our excellent principal investigators, with the resources to be able to get all 4 sets of those data, the XLRS data, the 2 different acrhomatopsia sets of clinical data, as well as at least the initial data for X-linked retinitis pigmentosa. So we feel we're very well positioned to use our resources to get this data and be able to move forward to deliver these products to the patients in need. Thank you for your time.